all of a sudden we've got scientists saying,
7 miles down, inside a titanium sphere barely wide enough to turn his shoulders, Victor Vescovo listened to the sound of metal contracting. The DSV limiting factor was descending at roughly 3 feet per second. And with every passing moment the ocean above was adding more weight to the water pressing against the hull. By the time the depth gauge reached 35,853 feet, each square inch of the sphere's surface was bearing the equivalent pressure of 8 full-grown elephants standing on it simultaneously. The viewports were thick acrylic, the diameter of a dinner plate. His breath was fogging the glass. The walls were cold. On the surface support ship above, a team of researchers watched a feed from exterior cameras mounted on the submersible. They were watching for what they expected to see: sediment, darkness, possibly some microbial film on the seafloor. The consensus view of Challenger Deep, the lowest point in the Mariana Trench and the lowest point on the surface of the Earth, was that it was essentially a barren environment. Too cold, too dark, too pressurized, too food scarce to support anything beyond microscopic life. Vescovo switched on the exterior lights, and the feed resolved from black into something that nobody on that ship had prepared themselves to see. Within 2 minutes, the assumption that had governed deep-sea biology for decades was gone. What was moving across that seafloor was not what the textbooks had described. And the more footage rolled, the further the scale of the error became apparent. Here is what the cameras found, why it was possible, and why the implications extend significantly further than a single research expedition at the bottom of one ocean. The environment that was supposed to be empty. To understand why the footage from Challenger Deep produced the reaction it did among the research team, you need to understand what the scientific consensus had been before the dive. The Hadal Zone, which refers to ocean depths below roughly 20,000 feet, was understood to be the most extreme environment on Earth. The pressure at Challenger Deep exceeds 16,000 pounds per square inch, compared to roughly 14 pounds per square inch at sea level. No sunlight has reached the floor for the approximately 4.5 billion years of Earth's existence. Temperature sits just above the freezing point of saltwater. The only food source reaching the bottom is what oceanographers call marine snow, a slow drift of dead organic material, fecal matter, and decomposed debris descending from the sunlit surface waters miles above. Based on these conditions, the scientific model was straightforward. Some microbial life certainly existed. Perhaps a few highly specialized invertebrates at the extreme margins of biological possibility. Nothing complex. Nothing active in any meaningful sense. Certainly nothing resembling a functional ecological community. The Five Deeps expedition was not primarily a biology mission. Victor Vescovo, a private explorer and retired naval officer, was attempting to reach the lowest point in each of the world's 5 oceans. A navigation and engineering challenge rather than a scientific one. The biological data collected during the Challenger Deep dive was, in a sense, a byproduct of having a very good camera at a place nobody had filmed with that resolution before. What byproduct it turned out to be. The first thing that appeared in the footage was amphipods. Crustaceans resembling shrimp, swarming around the bait package the team had placed on the seafloor, moving fast. Competing aggressively for access to the food source the way animals compete in any functioning ecosystem on Earth. Not a few cautious individuals picking at scraps. Clouds of them. And individual specimens measured several inches in length. For context, amphipods living in shallow coastal waters, where food is abundant and conditions are unremarkable, measure a fraction of an inch. The specimens at Challenger Deep were giants by comparison, thriving in an environment where nothing was supposed to support them at all. What do you think your first reaction would have been, watching that feed from the surface? Because several of the researchers on that ship reportedly stepped back from the monitor when the footage came through. Not in excitement. In a specific kind of disorientation that comes when something directly contradicts what you have spent years assuming was true. Fish with spines, 7 miles down. The amphipods, remarkable as they were not the finding that most directly challenged the existing framework. That distinction belongs to the snailfish. The snailfish captured on camera at depths exceeding 26,000 feet were 8 to 10 inches long. Translucent and soft-bodied, with small eyes, delicate fins, and clearly functional musculature. They were not drifting passively. They were hunting, swimming with evident purpose. Responding to the presence of the submersible and the bait package with the behavioral repertoire of active predators. Consider the biological implications of that sentence for a moment. A snailfish has a spine. It has a brain, a complete circulatory system, a liver, kidneys, eyes, a digestive system capable of processing prey. Every one of those organs is built from proteins. Proteins are molecules that function only when they maintain precise three-dimensional shapes. Pressure at significant ocean depth distorts and disrupts protein folding. At the pressures present at full hadal depth, the proteins that power every cellular process in a complex animal's body should not remain functional. And yet there they were. Not just alive. Not just surviving. Hunting. The explanation requires understanding what pressure actually does to biological material. The snailfish's body is primarily water, and water is essentially incompressible. The pressure outside the fish is matched by the pressure inside, cell by cell, in equilibrium. The animal does not feel crushed any more than you feel the 14 pounds per square inch of atmosphere currently pressing on your skin. You have simply never known a world without it. The actual challenge for deep-sea organisms is biochemical rather than mechanical. Proteins must maintain their functional shapes under conditions that tend to flatten and distort them. Cell membranes must remain fluid enough to allow molecules to pass through them even in near-freezing water. Every enzyme driving every metabolic process in every cell must continue functioning under pressure that would scramble the equivalent molecules in a shallow water animal. The solution these animals evolved is elegant in its simplicity. Their proteins have slightly different molecular geometries, stable under pressure where surface-adapted proteins would deform. Their cell membranes incorporate specific lipids that remain fluid at depth. Their enzymes are pressure-adapted variants of the same enzymes running in your own cells. And they carry a molecule called trimethylamine N-oxide, abbreviated as TMAO, which functions as a molecular scaffold, propping proteins into their functional shapes when pressure tries to collapse them. Shallow water fish have almost none of it. Measure TMAO concentration as you descend through the ocean, and it increases consistently with depth. At Challenger Deep, concentrations appear to approach a theoretical ceiling, which researchers believe explains why approximately 27,000 feet represents a natural depth limit for fish. Below that threshold, the molecule that enables life at depth runs out of room to do its job. Not exotic biology. Not alien chemistry. Small, specific refinements to molecular machinery that life has been using and adapting for billions of years. An ecosystem where there should not be one. The footage did not show one species or two. It showed a community. Multiple amphipod species with different body forms and distinct feeding behaviors. Several sea cucumber species using different locomotion strategies, some over a foot in length. Moving with evident purpose across the sediment surface. Multiple snailfish species adapted to different depth ranges within the trench. Polychaete worms. Isopods. Other organisms still being identified and catalogued from the footage at the time this research was being published. Layered across all of these were ecological roles. Predators actively hunting. Scavengers consuming what predators left. Filter feeders processing the water column. Organisms breaking down organic material in the sediment, releasing nutrients that other organisms then used. This was not a collection of isolated species barely surviving in separate niches. It was a food web, a structured community of organisms occupying distinct ecological positions and interacting across them. The same basic architecture as a coral reef. The same organizational logic as a temperate forest. Reproduced in complete darkness under pressure that should have prevented every link in the chain from existing. Here is the problem the footage created for the energy budget models. Marine snow, the organic material drifting down from surface waters, was calculated by researchers to arrive at Challenger Deep in quantities nowhere near sufficient to support the abundance and diversity the cameras documented. The math does not balance. Something else is feeding this system. Proposed candidates include chemosynthetic bacteria drawing energy from chemicals in the Earth's crust, converting geological energy into biological fuel the way photosynthetic organisms convert sunlight, unknown hydrothermal systems providing localized heat and chemical inputs, a recycling efficiency so high that almost nothing is wasted, more biological output per unit of organic input than any shallow water system achieves. Some researchers have proposed that the deep ocean may actually be more productive per unit of input than surface waters. That what looks like scarcity from above reflects a system of extraordinary efficiency rather than genuine poverty. If that hypothesis holds, the conventional picture of the ocean inverts entirely. The sunlit surface, volatile and seasonally unstable, becomes the marginal environment. The deep, cold, stable, and darkness-adapted, becomes the primary one. How would that change the way you think about the ocean? About which parts of it matter most, which parts are most worth protecting? And what we risk when we put things into the water without understanding where they go? The plastic bag and what it means. Among the footage from Challenger Deep, one image belongs in every honest account of this story. A plastic bag on the seafloor. 7 miles down, in the deepest place on Earth, in water that has been cold and dark for billions of years, there was a piece of human waste. And amphipod samples retrieved from the trench and analyzed in the lab contained microplastics in their digestive systems. The organisms at Challenger Deep survived 16,000 pounds per square inch of pressure for the entire evolutionary history of their lineages. They adapted to near-freezing temperatures. They solved the protein folding problem. They built functioning ecosystems in complete darkness. They did all of this across timescales that dwarf the entire existence of our species. They never evolved any response to what we put in the water. Because the contamination arrived before they had any time to encounter it, let alone adapt to it. The plastic bag had no natural precedent in that environment. The microplastics accumulating in amphipod digestive systems at full ocean depth arrived there because of decisions made on the surface. By people who either did not know or did not think about where the material was going. This detail sits uncomfortably alongside everything else the footage revealed. The discovery that Challenger Deep supports a complex, active, diverse ecosystem is remarkable. The simultaneous discovery that this ecosystem, in the most remote and inaccessible location on Earth, is already contaminated by human activity is a different kind of remarkable. We had classified this place as empty. It was not empty. We were putting things into it before we understood what was there. What stayed in the dark? Everything documented so far represents what was willing to be captured on camera. The amphipods approached the bait package. The snailfish investigated the lights and the unfamiliar presence of the submersible. The sea cucumbers were visible against the sediment surface in the illuminated zone. These were the organisms that had some evolutionary or behavioral reason to enter camera range. The footage also recorded things at the edges of what the lights could reach. Disturbances in the sediment outside the illuminated zone that had no clear source. Movement registering in the peripheral field of the cameras that resolved into nothing before the frame could capture it. Shadows at the boundary of the light that did not correspond to any species the team had documented on that dive. One camera operator reviewing footage frame by frame reported a repeated pattern of sediment displacement tracking the submersible from outside. Visible range, whatever was producing the disturbance keeping pace with the camera but never approaching it, for several minutes, before disappearing. None of this is conclusive evidence of any specific thing. Sediment disturbances have many possible causes. Movement at the edge of a light source is difficult to interpret reliably. Footage from an extreme environment reviewed by people already operating in a state of heightened attention generates patterns that may or may not reflect what they appear to show. What is on the record, and not subject to interpretation, is the sonar data. JAMSTEC, the Japanese Marine Research Agency, has conducted multiple surveys of the Hadal Zone since the early 2000s. Their acoustic returns include contacts that do not correspond to any species confirmed to inhabit those depths. Objects generating significant sonar returns, moving at depths where, before the limiting factor dive, nothing complex was supposed to be present. NOAA hydrophone arrays monitoring the Western Pacific have logged low-frequency signatures from the region that remain officially unclassified. These have been attributed to equipment artifacts and geological features. The framework for making those attributions depended on the assumption that nothing biological of significant size was living down there. That assumption is no longer available. The footage confirmed that complex, active ecosystems exist at full hadal depth. A snailfish hunting at 26,000 feet, a food web operating in complete darkness, these are no longer hypothetical. The sonar returns that have been logged and attributed to geological features were logged in an environment that science had, incorrectly, classified as biologically empty. Some of those contacts may be biological. Some of the objects generating them may be considerably larger than anything the cameras captured. Whatever they are, they have been in that darkness significantly longer than we have been sending instruments after them. The scale of what hasn't been looked at. The Challenger Deep footage covered a few dozen square yards of seafloor during a dive of limited duration. Everything described here, the amphipod communities, the snailfish, the food webs, the unexplained energy balance, the contamination, came from that small sample area. The full hadal depth range, from 20,000 feet to the bottom of Challenger Deep at 35,853 feet, spans approximately 45,000 square miles of seafloor distributed across trenches throughout the Pacific Rim. An area roughly the size of Pennsylvania. In permanent darkness. Under crushing pressure. Hosting, as the footage established, complex and active ecosystems that science only confirmed existed in 2019. Almost none of it has been filmed. The submersibles capable of reaching full hadal depth can be counted without using all the fingers of one hand. The number of dives to Challenger Deep conducted in all of human history is small enough to fit on a single page. The fraction of the hadal zone that has been directly observed by any camera is not measurable in meaningful percentages. It approaches zero. The implications of what the limiting factor cameras documented extend beyond deep-sea biology. The discovery that life can thrive in conditions this extreme and organize itself into functional ecological communities in complete darkness under pressure that was assumed to be prohibitive, dismantles the definition of the word uninhabitable in a specific and important way. Every environment that has been ruled out as too extreme, too crushing, too cold, too dark, deserves reconsideration. The oceans believed to exist beneath the ice shells of Europa and Enceladus, moons of Jupiter and Saturn, have been discussed as possible candidates for extraterrestrial life. Those discussions have always been cautious about the conditions those oceans would present: cold, dark, under pressure, disconnected from sunlight. The conditions that were supposed to prevent life from flourishing at Challenger Deep are structurally similar to the conditions those oceans would present. Life at Challenger Deep did not merely tolerate those conditions. It built a diverse, structured, active community in them, using molecular tools it refined over millions of years of exposure. The search for life beyond Earth has operated on the assumption that the conditions life requires are relatively specific. The footage from the deepest point on this planet's surface suggests the assumption is wrong in a direction we should find encouraging. Life doesn't need what we thought it needed. It needs what it can adapt to. And the evidence now available says it can adapt to almost anything. What does that possibility change about how you think about life's distribution in the universe? And what does it change about the priority we should place on understanding the part of this planet's ocean we have never seen? 7 miles down, in a darkness that existed before the first animal drew breath, something is moving. The cameras showed us the edge of it. Everything else is still dark. Share your thoughts below, because this is a conversation that extends well beyond any single research expedition. And the questions it opens are ones the scientific community is still actively working through.